Good afternoon, students. Good afternoon, everyone. Yes, good afternoon. Good afternoon. Yes, yes, good afternoon. Good afternoon. So far, we have completed the previous year question discussion for 2025 and 2024. Okay. Yes. Today, we are going to discuss for 2023 shift 1 question paper. Okay. In 2023, they have conducted in two different shifts, shift 1 and shift 2. Okay. Now, we are going to discuss for 2023 shift 1 question papers. Okay. Kindly be interactive in class. If you have any doubt, kindly message in the chat box. Okay. First question, what they are asking, they have given the different names of fatty acids, okay, whether it is saturated or unsaturated fatty acid, and they are asking whether it is an essential fatty acid or omega-3 fatty acid, omega-6, omega-7, like that they are asking. Kindly match and let me know. First thing, oleic acid. We know oleic acid will have 18 carbon and one double bond will be there. That will be at position 9. Petronocellic acid, where will be the double bond will be there in petronocellic acid? This is also 18:1 only, but the double bond will be shifted in position number 6. Clear? Gamma linolenic acid, in yesterday class itself we have seen, gamma linolenic acid means it will have double bond at position number 6, 9 and 12. Yes, 18:3, 6, 9 and 12. EPA, they are giving EPA, eicosapentaenoic acid. This is a typical example for what? Omega-3 fatty acid. This we have already seen in our class itself. This will have 5 double bonds. 5 double bonds will be there. Now you match. Yes, option D. Yes, we can see now. Okay. So oleic acid 18:1, del 9. Ninth position, double bond will be there. So this will comes under the class of omega-9 fatty acid. Yes. First option will come 4. So here only one thing is there. So this will be the answer. We will solve for other questions also. Petronocellic. Okay. Petronocellic will come as what? It's a omega-12 fatty acid. So option B. Okay. Clear. Then gamma linolenic acid. This will come as omega-6. 18 minus 12. So it will come as omega-C. Then EPA will come as omega-3 fatty acid. Can you tell any other example of omega-3 fatty acid? What are the examples of other omega-3 fatty acids? Yes. Alpha linolenic acid is one of the major example of omega-3 fatty acid. Yes, that will have double bond at position 9, 12 and 15. Then another most important thing is docosahexaenoic acid. Okay. This is another important omega-3 fatty acids. Okay. Docosahexaenoic acid. This will have 6 double bonds. Okay. 22:6. 22 carbon will be the 22 carbon length fatty acid chain with 6 double bonds will be there. The double bond will be there at position number 4, 7, 10, 13, 16 and 19. Clear? This is another important point. Then another important point I want to say here, omega-7 fatty acid. Omega-7 fatty acid. Example of omega-7 fatty acid is palmitoleic acid. Palmitoleic acid. Okay. We know this palmitic acid will have 16 carbon compound. Oleic acid will have 18 carbon compound with one double bond at position number 9. Okay. Carbon number 9. But palmitoleic acid will have 16 carbon with one double bond at position number 9. That's why we are calling this as omega 7 fatty acid. I hope this is clear. From this, sure they will ask one question. Okay. This is most important point. Okay. Next question. They are asking about histone octamers. Can anyone match and let me know? Nucleosome, a fundamental unit of chromatins containing a nucleosome core, which is composed of 146 base pair of DNA wrapped around the histone octamer. They are asking about histone octamer having which of the following composition? Okay. What are the composition of histone octamers? What will be the composition of histone octamers? It will have 2 copies of H2A, H2B, H3 and H4 histones will be there. Yes, correct. Yes. So, 2 copies of each of H2A, H2B, H3 and H4 will be there. That's why we are saying this as octamer. Okay, histone octamers. Okay, clear. Next. Yeah, yes, clear. Many can come correct answer. Now, they are asking the number of stereoisomers for N carbon aldoses. They are generally asking for aldoses and ketoses. What will be the number of possible stereoisomers? Okay, how to calculate that one? What is the general formula? Any guesses, students? What is the general formula for calculating the aldoses and ketoses? Yes. Aldoses means what would be there? For example, we can say glucose. Okay. Ketoses, example, what we can say? For example, fructose. This both will have 6 carbon. Correct. Yes, 6 carbon will be there. The general formula for calculating the number of stereoisomers for aldoses is 2 power n minus 2. This is very important. 2 power n minus 2. And for fructoses, 2 power n minus 3. Okay, 2 power n minus 3. If this is present in linear form, okay, if it is in linear form, this will have formula like this: 2 power n minus 2, 2 power n minus 3. If this aldoses and ketoses are present in cyclic form, the formula will change. Okay. If it is present in cyclic form, the number of stereoisomers present will be more. For example, 2 power n minus 1 will come as a formula for glucose. Okay. 2 power n minus 2 for ketoses. Clear? So, if cyclic form means the number of double bonds, then the number of possible stereoisomers will be more. They can ask, they will give the glucose, okay, what is the possible number of stereoisomers for glucose, like that they can ask. We know the number of carbon atom is 6 carbon in glucose. General formula is 2 power n minus 2. So, 2 power 6 minus 2, 2 power 4. So, total kitna? 2 2s are 4, 4 to 8, 16 possible isomers are there. Okay, this is clear. This is another important point. Okay, like this also they can ask the question. Okay, next question. They have given the different molecules, aspartate, glutathione, different molecules they have given. Then we have to match whether it is a pentapeptide or tripeptide, like that they are asking. Okay, whether it is a pentapeptide, dipeptide, like that they are asking. Any guesses? Yes, any guesses? Aspartame, it is what? It is an artificial sweetener. Yes, it is an artificial sweetener made up of exclusively of L-phenylalanine and L-aspartate. This we have already seen in our class itself. Yes. So, phenylalanine and aspartate. This is what? Dipeptide. Okay, this is a dipeptide. So, A will come where? A will come option 4. Okay. For A, option 4. Second, glutathione. Yes, glutathione. We have already seen ECG. It is a what? It's a tripeptide. So, B will come option 1. Okay. B will come option 1. Oxytocin is a typical example for nanopeptide. So, okay. Nanopeptide means what? 9 amino acids will be there. Clear? So, C will have 3. Endomorphin, this is another important example. Endomorphin is a cyclic tetrapeptide. Okay. Endomorphin is a cyclic tetrapeptide. Okay. So, option B will be the right. Excellent. Excellent. B is the correct answer. Then, Before going to next question, I just want to say some important things. Okay. Here they are asking about DESP peptides. Okay. What, sir, peptides, we know what is this DESP peptide. Okay. DESP peptides means nothing. We know in each amino acid, for example, this is one amino acid. Okay. This is one amino acid. Then another amino acid will join with what? Peptide bonds will be there. Okay. So another amino acid will be joined by peptide bond. Okay. C double bond O N H C alpha carbon. Okay. This we will say as peptide bond. Okay. This peptide bond will be always in what? This will be in transform. Okay. Trans. This O and H will be present in opposite direction. Transform. Then another important thing is what? This will have partial double bond nature. This also clear na? So partial double bond nature will be there. Because of this, the rotation will be not flexible. Okay. Only certain pi and psi values will be possible. Okay. Only certain pi and psi value will be possible. This will be involved in what? Secondary structure formation. Okay. This is normal peptide. If DESP peptide, what it will happen means this CONH peptide bond will be converted into ester bond. Okay. This will be formed as ester bond. First point is that will be formed as a ester bond. And second most important thing is there will be a easy flexibility for rotation of the C alpha carbon. Okay, clear. So DESP peptide means first thing peptide bond will be replaced by ester bond. Second most important thing, there will be a easy flexibility will be there. Okay. Now you solve this. Okay. DESP peptides have DESP peptide have more flexible structure, lower rotational barrier for cis trans isomerization than their native analogs. Whether this is true or not? Yes, we have seen this is true. Second reason what they are saying means one or more amide linkage. That means what? Peptide bond in DESP peptides are replaced with corresponding ester groups, leading to their reduced hydrogen bonding capability, which in turn result in deformed secondary structure. Yes, both assertion and reason are correct only. So, option A is the right answer. Clear? So, both A and R are true and R is the correct explanation for A. Okay. And yes, very good, correct. Now, sixth question, what they are asking, see here, they have given the directly the answer. See, old yellow enzyme. The enzyme is yellow. Okay. Yellow enzyme is there. Read the question properly. It's a yellow enzyme isolated by Warburg and Christian. Okay. From Breves botanist in 1932. Okay. What they are asking means it's comprised of colorless apoprotein. Okay. And a yellow cofactor. The key point from this question is yellow cofactor is there. Okay. Yellow cofactor is there. We know vitamins will act as a cofactor for various chemical reactions. Okay. In our vitamin class itself, we have already teased vitamin riboflavin. Take over. Riboflavin. That is vitamin B2 will be yellow colored due to present of this ribitals groups. Okay. This we have already seen. So FMN and FAD are the typical cofactors. Okay. Typical cofactors. Yes, correct. So the answer is what? Flavoenzyme. The yellow color is imparted due to the presence of that vitamin D cofactor. So flavoenzyme. Okay. Next, they have given the difference. See, from every year they are asking one question from enzyme classifications. Okay, enzyme classification. They are giving the different chemical reactions or different enzyme names, and they are asking to match with this class of the enzyme. Okay, just match this first. Formation and removal of carbon-carbon double bond. So carbon-carbon double bond is formed or removed. Okay, first thing is what? It will come as lyase. Yes. So where only one option they have given, option A is 3. Yes, B is the right option. Yes, B. Yes. So lyase. Amino acyl tRNA synthetase, what it will do? It will bind the amino acid by utilizing amino acid with the tRNA by utilizing the energy. So ATP will be utilized there. Yes. So it will come as a class of what? Ligases. ठीक है, transamination, only the amino group will be transferred from one amino acid to other, ठीक है, one amino acid to other alpha keto acid. So this is a transferase type of reaction. So this is B. Removal of fatty acid from triacylglycerol. Here what? It's a digestive enzyme. It will come in the class of hydrolase. So the right option is option B. Yes, correct answer. Uday Lakshmi, correct. Yes. Next, simple question. This is repeated question. You have to see here. The catalytic center of which of the following protease family consists of catalytic triad of aspartate, histidine and serine? Any guesses? What will be the answer? What will be the answer? Whether it is a cysteine protease or aspartate or serine protease or metalloprotease. What will be the answer for that? Whether it is, yes, yes, excellent. It's an serine protease. Okay. Can anyone tell? Yes, serine protease is the correct answer. Can anyone tell HIV protease is an example for which type of protease? HIV protease, pepsin, renin. These all are example of which type of protease? This we have already discussed in our previous year question paper discussion. This they have asked in 2023. Sorry, 2025. What will be the answer? HIV protease. It's an aspartic acid protease. That's what we have. Yes, aspartic acid protease. Okay. Then another most important thing what they can ask from this question is, what is the specific aspartate, histidine and serine residue present in the catalytic triad of this serine protease? Okay. Serine 195, histidine 57, aspartate 102. Okay. This is another most important point to be remembered. Okay. Serine 195, histidine 57 and aspartate 102. Okay. Next, simple thing. This you will answer. I know. Okay. The first three step of beta oxidation pathway of fatty acyl CoA. First three step of beta oxidation of fatty acyl CoA chemically resemble the citric acid reaction carrying out which of the following biochemical conversion? First, we have to know beta oxidation, first three pathway. What will happen? First, fatty acyl CoA will be there. Okay. Then acyl CoA dehydrogenase enzyme will act. Here, FADH2 will be produced. Yes. This is clear only. Then here, enoyl CoA will form. Enoyl CoA hydratase will come. Here, water molecule will be added. Okay. Here, beta hydroxy acyl CoA form. Here, another dehydrogenase enzyme will come. That will be lead to production of NADH. This is clear for everyone. I know this will be clear. Yeah. A similar reaction. If you see in TCA cycle, there will be only one place where FADH will be produced. That is what? Succinate dehydrogenase enzyme only will produce FADH2. Succinate dehydrogenase will convert succinate to fumarate. So the first product, first substrate starting is succinate. You see the answer. So succinate to oxaloacetate. So these two reactions are similar like that they are saying. Okay. So yes, succinate to oxaloacetate. Yes, yes, very good. Next, another important question from beta oxidation they have asked. During our fatty acid catabolism itself, I have teached all these things. Just match and let me know. They have given 2,4-dienyl-CoA reductase, different enzyme, phytenyl-CoA hydroxylase, acyl-CoA oxidase, cytochrome P450. This is omega oxidation. Okay. Now you match and let me know the specific enzyme for unsaturated fatty acid degradation. What are the enzymes? Two enzymes will come extra. That we have discussed in our class itself. Yes, match and let me know. 2,4-dienyl-CoA. Yes, 2143. 2143. Yes. 2,4-dienyl-CoA. That will be specifically involved in the oxidation of unsaturated fatty acid. Yes, it is involved specifically in oxidation of unsaturated fatty acid. Apart from that, what is the other enzyme? 2,4-dienyl-CoA reductase and del3-del2 enoyl-CoA isomerase. Enoyl-CoA isomerase. This they asked in 2025 question. Okay. 2025 they have asked about this isomerase. Okay. Phytanyl CoA hydroxylase, that will be specific for alpha oxidation. This we have seen in our class. Acyl CoA oxidase, this will be there in peroxisomal beta oxidation. So this will come as C. P450, it's a mixed function oxidase enzyme that will be involved in omega oxidation. So this will be the right option. Yes, very good. Next, they are giving about inhibitor in glycolysis. Okay, this also I have discussed. Okay, inhibitor of glycolysis. Okay, assertion: the addition of fluoride to the fermenting yeast extract causes build-up of 3-phosphoglycerate and 2-phosphoglycerate. If fluoride is added, that will be lead to the accumulation of 3-phosphoglycerate and 2-phosphoglycerate. So this is ultimately inhibiting some of the enzyme. Okay. The fluoride strongly inhibit the enolase enzyme. They are saying enolase enzyme is inhibited by forming tightly bound complex with magnesium at the active site. What will be the answer? Whether assertion and reason are correct? Whether reason is the correct explanation of A? Any guesses? Yes, any guesses? Yes, excellent. See, this fluoride is a typical inhibitor for enolase in of glycolysis. Okay. If enolase is inhibited, there will be a no formation of phosphoenol pyruvate. So this 3-phosphoglycerate and 2-phosphoglycerate will be accumulated. Okay. How this fluoride will go and inhibit the enolase means that will go and bind with the catalytic site. Okay. Then the magnesium binding will be inhibited because of this complete reaction will be inhibited. So this is right. Yes. Next, another important question from glycolysis. See, two questions they are asking continuously from glycolysis. Okay. Second question. During glycolysis, the enzyme phosphoglycerate mutase. What it will do? It will convert 3-phosphoglycerate to 2-phosphoglycerate. Yes, this is the reaction. Catalyze the conversion of 3-PG to 2-phosphoglycerate through formation of, while converting this 3-PGA to 2-PGA, there is an side chain reaction that will be lead to formation of 2,3-bisphosphoglycerate. Okay, 2,3-bisphosphoglycerate. Okay, this is the question. Okay, the phosphoryl group, for example, here only one phosphate will be there, 3-phosphoglycerate. Here also only one phosphate will be there. But in the side chain reaction, in the other thing, 2,3-bisphospho, there is 2 phosphate is there in the product. Where come this second phosphate came? Whether it came from ATP or acetyl phosphate or pyrophosphate, or whether the enzyme itself is donating the phosphorus to that compound, like that, they are asking this question. This is another important question. Okay, another important question. Okay, this will be specifically happen in what? Erythrocytes. Okay, in erythrocytes, this will be specifically happen. Why means this 2,3-bisphosphoglycerate will act as an what? It's an inhibitor. Okay, it's an inhibitor, or it will decrease the affinity of oxygen for hemoglobin. Clear? Oxygen affinity for hemoglobin will be decreased. So the correct answer for this what? The enzyme itself will have the phosphate group, because the enzyme will be active only after the phosphorylation. The active phosphate group from the enzyme will be donated to the substrate, that will be lead to the formation of 2,3-PGA. Okay. Most important thing, this cycle name is what? That is known as Rapoport-Lewis cycle. Okay. This will be specifically happen in erythrocytes. Okay. See here, this is the reaction, 1,3-bisphosphoglycerate, 3-phosphoglycerate is happening. Okay. See here, 2,3-bisphosphoglycerate will be formed. Okay. Clear? This is very important point. Okay. If you want to work for more objective questions, and if you want to practice more objective questions and topic-wise mark text, kindly take this course. Okay, this will be very useful, very much useful for all of you. Okay, then those who are targeting for ICAR examination 2027, they can kindly enroll in their plant biotechnology course. Okay, they can enroll. First 100 students will get, and offer also is going on only for first 100 people who are enrolling in their course. Okay. Next, another important thing, they are particularly asking about pyruvate dehydrogenase complex. They are asking about pyruvate dehydrogenase complex and different cofactors. Okay, different cofactors involved in pyruvate dehydrogenase complex. This is another simple question. I know you will answer. Okay, NAD. This is NAD. Okay, NAD. Nicotinamide adenine dinucleotide. Okay, where it will come as a cofactor? Lipoic acid, CoA, FAD. They have given the different cofactors. Now we have to match which one is substrate for E2, substrate for E3, where this cofactor will bind, like that we have to match. Kindly match and let me know. Yes, this we have clearly discussed in our lecture itself. Yes, anyone can match and let me know. Yeah. Nicotinamide adenine dinucleotide. Where it's coming? It's coming as a substrate for enzyme 3. Yes. So A4, where A4 only one option is there. This will be the right option. Okay. A4. Then B, lipoic acid. Where it is bounding? It is bounding to, it is covalently bounding to lysine residue. See here, lipoid will be binded here. So this will be the right option. CoA, then FAD. Okay, this is very simple question we have discussed in our class itself. Okay, clear students? Next, different inhibitors of electron transport chain. Different inhibitors of electron transport chain, and they have asked which inhibitor will go and inhibit which complex. Okay, we know typical example, pyridine, amytal, and rotenone. This will specifically, it will go and inhibit the complex one. Okay, complex one, what it will do? It will takes the electron from NADH. So NADH dehydrogenase complex is complex 1. So amytal will specifically inhibit the NADH linked oxidation. So this will be correct. So A will come option 2. Where it's coming here only, it's coming, A will come option 2. Yes. Then we can see whatever things important, like simple one, acid. Acid, what it will do? It will go and inhibit complex 4. Yes, acid is the inhibitor for complex 4. Whether anything is matching here, yes, completely inhibits oxidation of electron donor. Yes, this is another important thing. Okay, so D will come option 3. Okay, D will come option 3. Among this, only this one have correct answer. Okay, so D will come option 3. See here, for A, to inhibit NADH complex. B, antimycin, FADH complex will be there. TMBT, it's an inhibitor. What it will do means, it will transfer the electron directly to the cytochrome C. Okay, this is simple question. Okay, just you have to select the option by elimination method. Okay. Next, Randall cycle. Sir, what's the new terms you are saying? Randall cycle. Okay, Randall cycle. This Randall cycle, we can also say this as what? Glucose fatty acid cycle. Okay, glucose fatty acid cycle. We know after fatty acid oxidation, acetyl CoA molecules will be produced. Okay, acetyl CoA molecule will be produced. This acetyl CoA will bind with oxaloacetate, that will lead to the formation of citrate. Okay, this citrate, what it will do means, it will go and inhibit the glycolysis. Why? If glycolysis happen means, ultimately acetyl CoA will pyruvate will form, that will be converted into acetyl CoA, that will enter into electron transport chain for producing energy. Already beta oxidation is happening, so glycolysis no need of happening. That's why it's stopping the pathway. How? This citrate will go and inhibit the phosphofructokinase 1 enzyme. We already discussed this. PFK1 is the major regulatory enzyme for glycolysis. So citrate is an inhibitor for that enzyme. It will go and inhibit the specific step of the glycolysis. Because of this, glycolysis will not happen. This is known as Randall cycle. Okay. Now you answer. The Randall cycle describe the inhibition of glycolysis by fatty acid oxidation. Yes, this is true. Oxidation of fatty acid increase the concentration of citrate that inhibit phosphofructokinase enzyme of glycolytic pathway. This is also true. So Right option is what? Yes, option A. A and R are true, and R is the correct explanation for A. Next, simple thing, another thing, the chemical identity of oxygen evolving complex, oxygen evolving center present in PS2 of plant and cyanobacteria is, what will be the answer? Presenting in plants and cyanobacterial. The chemical identity of oxygen evolving center present in photosystem 2 of plant and cyanobacteria is what? Any guesses? What will be the answer? What will be the answer? Yes, option A. No. Any guesses? Yes. The right option is what? Metal oxygen clusters. TK metal. We know in plants, magnesium and chloride will be act as a clusters, or else magnesium calcium oxides, this all calcium monoxides, this also will act as an clusters that are involved in electron transport chains, okay, of oxygen evolving complex. This is very important point, okay. Another important thing, the energy efficiency in the number of ATP equivalent per absorbed photon of non-cyclic electron transport process in photosynthesis after taking into the ATP yield by NADH. This is another simple thing. See, if non-cyclic electron transport chain is happening, means this is PS2, this is PS1, okay. Total 2 photons will act on this, another 2 photons will act on this, total 4 photons will come. Per molecule of oxygen, per molecule of H2O is degrading, okay. But usually if you see, for evolving 1 molecule of oxygen, we need 8 photons, okay, 8 photons, 8 to 10 photon is needed, okay. So 8 to 10 photons are needed per molecule of oxygen. This is clear, 8 to 10 photons are needed. If you see the ATP yield, there will be what? There will be 3 plus 1, okay, 4 ATP yield will be there. Then 2 NADPH will be produced at the end of the reaction. So 2 NADH and 4 ATP, okay. So 4 plus 2 into 3, kitna 10 ho gaya, okay. So total of 10 will be there for 8 photons. So 10 by 8, what it will come? 1.25, okay. This is just a simple calculation. You just go through this one, okay. Next, another important thing they are asking from photosynthesis only, they have given different enzymes involved in photosynthesis, then with glycolysis, okay. Two important things, two different enzymes. One is pyruvate kinase. One is pyruvate kinase. What it will do? It will catalyze irreversible step, last step. Phosphoenol pyruvate will be converted into pyruvate, okay. Another thing they are saying in our question, pyruvate phosphate dikinase. Pyruvate phosphate dikinase, dikinase. This will lead to the formation of what? Phosphoenol pyruvate and PPI, This is what? This is a reversible reaction. This will happen in C4 cycle. This we know already, Now you observe the question properly. Pyruvate phosphate dikinase is a key enzyme of photosynthesis. Yes. And catalyze a similar reaction to one catalyzed by pyruvate kinase, a glycolytic enzyme. Which of the following statement about these two enzymes is true? They are asking which will be true. Now you listen. Pyruvate phosphate dikinase catalyze an irreversible reaction. Whether it is true? No, it will catalyze a reversible reaction. Next, pyruvate kinase catalyze a reversible reaction. This is also wrong. It will catalyze irreversible reaction in glycolysis. Next, pyruvate kinase consume 1 molecule of ATP. Here they are saying pyruvate kinase is consuming ATP. No, it will produce ATP. It will produce ATP. So this will also will not come. And what is the leftover option? Pyruvate phosphate dikinase consume 1 molecule of ATP for each molecule of pyruvate convert into phosphoenol pyruvate. Yes. So this will be the right option. Next, they are giving about how the nitrogen metabolism. In nitrogen metabolism, how the nitrate reductase enzyme is regulated. This we know by phosphorylation. If the nitrate reductase is phosphorylated, that will be inhibited. This we have already seen. This phosphorylation will be specifically happen at serine residue that will be present at hinge 1. This also we have discussed in our class. H1, hinge 1. Okay. Now we will see the questions. Okay. Which of the following region of nitrate reductase enzyme is extremely important for 1433 protein mediated post-translational regulation of its activity? We know if the enzyme is phosphorylated, this 1433 protein will come and bind here. So this site will be common. Okay, the serine phosphorylation site is common to this 1433 binding protein. We already discussed. This is where it will happen. It will happen at H1. Okay, it will happen at Hing1 region between molybdenum containing cofactor and the heme domain. This we have already discussed in our lecture. Okay, clear? The synthesis under activity of nitrogenase enzyme. Last question they have asked about nitrate reductase. Okay, nitrate reductase. Here they are asking about nitrogenase enzyme. Get stimulated by which of the following condition? Okay. Get stimulated by which of the following condition? Okay. They have given different condition. High oxygen concentration. It will ultimately inhibit the nitrogenase activity. High expression of NiFL. What this NiFL will do? It's an inhibitor of other NiF gene expression. So this also will not come. So NiF A will be an activator, that they have never given our option. Then higher expression of DRAT enzyme. Okay, DRAT. Dinitrogenase reductase ABC ribosyl transferase. In 2024 question, they have given this one. Okay. Nitrogenase will be regulated by what? By ADP ribosylation. Okay. If ADP ribosylation will happen, means this nitrogenase will become inactive. In our question, they are asking in which condition it will be active. So this also will not come. If low glutamine is there, means what? Ultimately, ammonia content in the cell is less. This nitrogenase, what it will do? It will convert atmospheric nitrogen to ammonia. So if low glutamine and high concentration of alpha glutamate is there, means nitrogenase activity will be enhanced. Yes, clear. Which of the following amino acid in protein or peptide bond does not contribute significantly toward UV absorption at 280 nanometer? Okay. We know proteins will absorb at 280 nanometer. DNA means 260 nanometer. This we are clear. In 280 nanometer, particularly aromatic amino acids, amino acid will observe this UV. Yes. Particularly, yes. Kencom, correct answer. Yes. So they are asking in our question which amino acid will not observe. So cysteine will not observe. So tryptophan, tyrosine and phenylalanine will observe. If you see the order, tryptophan will observe more, followed by tyrosine, followed by phenylalanine. Okay, this order also you have to remember. Okay, clear? Next, they have asked from technique in biochemistry. Okay, technique in biochemistry question. Okay, they are asking about chromatography. You see here, which of the following intervention would result in enhanced chromatographic separation? How to increase the efficiency of the chromatographic separation? How to get a clear peak? Okay, how to get the increased separation? Okay, this is another important thing. See here, first option they have given increasing the number of theoretical plates. If n is increased, this is clear. If number of theoretical plate is increased, means the equilibrium of separation will be more. So we will get an enhanced separation. So option A will correct only. Okay, option A is correct. Second, decreasing the height equivalent to a theoretical plate. There is one correlation. Okay, number of theoretical plate equal to L by H. Okay, H is what? H is height equivalent to the theoretical plate. So if we decrease this one, number of theoretical plate can increase. So ultimately it will enhance the chromatographic separation. So what? So that will come. Excellent, can come option A. Very good. We will solve this. Okay. Third thing, decrease in the column height. This is ultimately wrong. If you decrease the column height, it will decrease the efficiency of the chromatographic technique. Okay. So column height should always increase. Then another important thing, decreasing the size of particle used to pack a column. So it will also increase the efficiency. So the right option is A, B and D. Already people has messaged A, B and D. Yes. Another important thing from basic biochemistry. This year they asked around 60 questions from biochemistry only. Okay. Related to biochemistry only. So listen properly. Okay. Read properly. They have given the different lipid names. Name of the lipids they have given. Then we have to match how the structural significance is there with this structure. What is ceramide, cerebroside, gangliosides, platelet activating factor, everything we have discussed in our classes. Okay. Ceramide means what? This will the specific backbone for sphingolipids. Yes, it's a structural parent of all sphingolipids. So A will come option 3. Cerebroside, if with the ceramide one sugar is there, that we will call as cerebroside. Okay, so that is what? That is a neutral glycosphingolipid. Yes. So B will come 4. Where B is coming 4? B is coming 4 in both of these things. Okay. B will come 4. Ganglioside. This in our class itself we have discussed. It will have sialic acid residue, because of that negative charge will be there. Okay. That is acidic charged glycosphingolipid. So C will come 2. So this will our right option. Okay. Platelet activating factor. These are example of ether linked glycerophospholipids. Okay. Ether linked platelet activating factor is a typical example for ether linked glycerophospholipid. Okay. Another thing they are asking about allosteric enzymes. One question from enzymology. Okay. Some of the exceptional properties of regulatory enzyme that are allosteric enzyme. We know allosteric enzyme means what? This will not obey the Michaelis-Menten constants. This is the first thing. Second thing, this will be what? Multimeric enzyme. There will have different substrate binding sites. Okay. Different sites will be there for substrate binding. Then it will show phenomenon of cooperativity. Okay. If a substrate is binding one place, then if a modulator is binding at the another place, that can influence the binding of other substrate by the action of cooperativity. Okay. What they have given? Their kinetics do not obey the Michaelis-Menten equation. Yes, this is true. They are mostly monomeric. No, this is what? Multimeric proteins. More than one subunit will be there. So B is wrong. Okay. Second, they have more than one substrate binding site, and the substrate binding site to different site is mutually independent, exclusively and non-cooperative. They are given non-cooperative. I already told it's a cooperative expression will be there, so C also will not come. Binding of effector molecule to them can lead to their activation or inhibition. This is correct, yes. So option D will come. Yes, this is correct. Next, different cofactors they have given. We have to match with this coenzyme. With vitamins with that coenzymes. Okay, vitamin with that coenzyme. Pantothenic acid, para-aminobenzoic acid, ribose, pentonic acid. Okay, we know PABA, para-aminobenzoic acid is a para-aminobenzoic acid along with glutamic acid. This is the critical precursor for what? Folic acid synthesis. In our vitamin class itself, we have seen. So, B will come option 3. You see, this is the simplest thing. Where B is coming option 3. Okay, nowhere it's coming. Only one option is coming, B. Okay. Now, we will solve for other questions. Pantothic acid. Pantothic acid will be present in what? Coenzyme A. Okay. Ribose will be a component of NAD. Pentonic acid will be there in lipoic acid. Okay. This is clear. This is simple question only. Okay. They have given different vitamins. We have to match with its coenzyme. Okay. Another question. Different minerals they have given here. Different minerals they have given. We have to match with that deficiency symptom. Okay. Among this what? Selenium. This is specific enzyme. Specific mineral nutrient present in what? glutathione peroxidase. Okay, this is very much important enzyme to maintain the redox state of a cell. Okay, if redox state of the cell is not maintained, okay, that may lead to oxidative stresses. Okay, so first thing is what? Enhanced oxidative stress. So B will come option 3. Only one thing is coming. See, selenium enhanced oxidative stress. B. Okay. Manganese. If manganese is deficient, it will inactivate the prolidase enzyme. Okay. Prolidase enzyme will be inhibited. Okay. Magnesium, what it will do? It will poor vitamin D status will be there because all the enzymes that are involved in vitamin D synthesis will require magnesium as a cofactor. Okay. Molybdenum, if molybdenum is deficient is there, means neurological abnormalities is there. Neurological abnormalities will be there. Why? Means due to sulfide oxide deficiency, because that will act as a cofactor for this sulfide oxide enzyme. Clear? Next, photosynthesis question. Solve one, let me know. Photosynthesis. Given below are two statements. One is assertion and another is label as reason. Yes. Any guesses? Any guesses, students? Camp plants undergoes nighttime acidification and daytime deacidification. Nighttime will be acidified, daytime will be deacidified. Yes, it's true. Nighttime stomata will be open, CO2 will come, malic acid accumulation will be there. Daytime, malic acid will be decarboxylated. Yes, this is true. Camp plants take up CO2 during daytime. No, this is wrong. So A is true and R is false. Okay. Yes. Next, about amino acids, histidine. About amino acid histidine, we already discussed in basic class itself. Histidine is the specific amino acid that will have a pKa near to physiological pH. Okay, near to physiological pH. The free histidine is a dominant buffering system at physiological pH. Then the protein bound one dipeptide histidine. You see, if the histidine is free, there will be amino group will be there. This will have partial positive charge. Then R group will be there. That will also will have amino group. Okay. This will have certain pKa. Okay. This pKa will be helped in buffering system. Okay. But if this will be present in peptide bond formation, this amino group will be involved in peptide bond formation. So what? This repulsion between the amino group will be reduced. Because of that, what it will happen? It will increase the pKa. It will increase the pKa, so that will have a better ability to do the buffering system. Okay. This concept is clear. So, free histidine is a dominant buffering system at physiological pH. Then the protein bound or dipeptide histidine. Yes, this is true. In protein or dipeptides, the imidazole pKa decrease substance. Why? No, it will increase. Correct? It will increase this thing. So, A is true and R is false. This is correct option. Bundle seed cell. They are asking what is the characteristics of bundle seed cell in C4 plants. Characteristics of bundle seed cell in C4 plants. Have thin. They are saying thin cell walls will be there. Centrifuged arranged chloroplast. Yes, this is correct. This is correct. So wherever option B is coming, that will come. Okay. Contains stacked thylakoids membranes and do not have starch granules. This is wrong because why? In bundle seed cell only, what will happen? CO2 fixation will be happen at last by C3 cycle in C4 plants. Okay. There is an starch granules will be there. So wherever C is coming, this will not come. Okay. Carry out light independent reaction of photosynthesis. Yes. Wherever D is coming, that will come. Have thick cell wall. This is also true. Okay. So A, B and D will come. Okay. Students, they are asking here thin, not thick. How it will come? If it is thick cell wall, it will come. Okay, so B and D will be the right option. Okay, there are another question they are asking about Kennedy pathway of lipid biosynthesis. Kennedy pathway of lipid biosynthesis. Just match and let me know. Yeah, Kennedy pathway of lipid biosynthesis. Okay, which of the following metabolites is not intermediate in triacyl glycerol biosynthesis by Kennedy pathway? Yes, glyceraldehyde 3-phosphate will not come. We know lysophosphatic acid will come. Here fatty acyl transferase enzyme will come, that will lead to phosphatic acid. If phosphatic acid will be treated with phosphatase enzyme, that will lead to formation of diacylglycerol. So the right option, which is not coming in the pathway. So glyceraldehyde 3-phosphate will not come in this pathway. Yes. Next, in a nucleotide sequence, ATGC, they are given ATGC is the sequence. Which of the following nucleotide has unlinked 5' hydroxy? They are saying which will have unlinked 5' unlinked 5' hydroxy group. If you see, this C will have 3' hydroxy group. Okay, this C will have 3' hydroxy group. This 5' will have unlinked 5' hydroxy group. Clear? So the right answer is what? Deoxyadenylate will have 5' hydroxy group. 3' hydroxy group will be present in deoxycytidylate. Clear? So this will be present in 3' 3' hydroxy will be present in deoxycytidylate. Okay, clear? Which of the following subunit of RNA polymerase complex is absent during elongation phase of bacterial transcription? If bacterial transcription is happening, which of the following RNA polymerase complex will be absent? See here, which of the following? See, sigma factor. The major function of sigma factor is what? Just for promoter recognition. It has to go and recognize the promoter. Okay. Once the promoter is recognized, it will initiate the transcription. If transcription is initiated, this sigma factor will remove from the enzyme. So, due that time, this sigma factor is not needed. Okay, clear? Yes. Next, which of the following is not related to termination of transcription in E. coli? Okay, termination of transcription in E. coli. We already discussed in prokaryotics, transcription may be terminated by two ways. One is row dependent and row independent pathway. Okay. Row protein is there, this will come. Okay. Intrinsic terminators, that is row independent, that will also come. If you see this N protein, this N protein will be particularly involved in where? Means bacteriophages. Okay. Bacteriophage, particular bacteriophage lambda it will come. So, this will be the right option. Okay, they are asking which will not be involved. NUSA will become, okay, this will be involved in transcription elongation. During elongation stage, this NUSA protein will involved in E. coli transcription. Okay, so the right option is N protein. Okay, because that will be present in bacteriophage lambda. Okay, what is iso-accepting tRNA? What is meant by iso-accepting tRNA? Any guesses? Iso-accepting tRNA. We know there are 64 codons are there. Among them, 61 will be coded for amino acid, 3 for stop codon. Okay. If you see in this 61, it is going to code for only 20 amino acids. Okay. So, only 20 amino acid will be coded by this 61 codons. If you see here, yes, C is the correct answer. Yes, C is the correct. If you see here, this 20 amino acid will be coded from more than one codons. Okay. So, the correct answer is what? Different tRNAs that are specific for the same amino acid. Okay. So, different tRNAs will be there that will be specific for same amino acid. Okay, clear? For example, tRNA 1, tRNA 2, 3, 4 is there. This all is coding for arginine. Okay. Like this. Okay. Arginine, it will code. Okay. R is the symbol for arginine. Okay. A for alanine. Okay. Clear? So, C will be the Right option. Okay. The consensus sequence 5' ACC AUUC is also known as what? First you have to remember here. 5' ACC AUGGG 3'. What will be the right answer? Yes, any guesses? What will be the right answer? It's Cossack sequence. Okay, it is Cossack sequence. This is direct question. It's Cossack sequence. Then child organ sequence means what? It will be a purine rich that will be involved for what? That will be involved in binding of ribosome to mRNA, particularly in prokaryotics. This they asked in 2024 question. We have seen this one. Yes, we have seen this one. Next, which of the following is type 2 restriction enzyme? Simple hind 2, that is the first restriction enzyme. Then that is the typical example of type 2 restriction enzyme. Type 1 will come what? HNIF3, ECO-K and ECO-KB will come in type 1 and type 3. Okay. Which of the following is the activity of reverse transcriptase? Reverse transcriptase, what it will do, students? What it will do? Reverse transcriptase. It will synthesis the cDNA from mRNA. Yes, it will synthesis cDNA from mRNA. Okay. Yes, that is correct. Yes. Yes, synthesizing cDNA from mRNA. Mani is very active in the class. Very good. P blue script is an example of P blue script is an example of. This is typical example for parchment vector. This we have seen. This will have FAS. FAS site will be there. Okay. Then another thing, it will have a plasmid resistant gene. That means col1 fragments will be there. Okay. Clear? Which of the following chemical is used to make E. coli cell competent to take the external DNA? This you guys have to answer. This we have already seen in our recombinant DNA classes. Recombinant DNA technology, I have mentioned this one. Yes. Which chemical is used to make E. coli competent to take the external DNA? Sodium acetate, SDSPH, calcium chloride. Yes, excellent. Calcium chloride. We will use calcium chloride. Some cases magnesium chloride also they will use. So major is calcium chloride. Yes, very good. Northern blot hybridization, which will be immobilized on the membrane, whether DNA or RNA or protein, which will be immobilized on the membrane. Whether RNA will be immobilized or DNA, what? Which technique will be used for studying the copy number of transgene characterization? Yes. So RNA sample is immobilized on the membrane. Okay. In northern blotting, RNA sample. DNA means southern blotting. Okay. Protein means what? Protein means western blotting. Western blotting means protein. Okay. Then another important question. Copy number of transgene can be known. Copy number can be known by what? Southern blotting. Okay. This also can be done by southern blotting. Yes. Which of the following structure is commonly found in both prokaryotic and eukaryotics? They are saying some structures are there that will be present in both prokaryotic and eukaryotic, whether centromeres or ribosomes, vacuoles or mesosomes. Vacuoles will be not present in both organelles. Ribosomes, yes. 70s will be there in prokaryotic, 80s will be there in eukaryotic. So the right option is ribosomes. Which of the following is not a part of endomembrane system? Yes, endomembrane system. Endomembrane system will be there like this. Okay, endoplasmic reticulum will come. This will be connected with Golgi. Okay, if cell has to be connect, this will be act as a plasmodesmata. So what will be the right answer? Yes, peroxisome will not come as an part of endomembrane system. Yes, very good. If half of the progeny of a test cross are having recessive trait, they are saying test cross, half of the progeny is recessive. What will be the genotype of the dominant parent? They are asking what will be the genotype of the dominant parent from genetics. Anyone can answer. What will be the genotype of the dominant parent? whether it will be homozygous dominant or heterozygous dominant. Trait is governed by multilocus or trait is epistatis. Yes. What will be the answer? Homozygous dominant, heterozygous dominant. Trait is governed by multiloci. Trait is epistatis. Yes. Heterozygous dominant is the right option. Yes. Very good. Swiss Prot, repeated question. 2025 they have asked. 2024 also they have asked. Okay. Now they are asking. They asked in 2023 part 1 also they are asking. Okay. Swiss Prot is a specific for what? It's a secondary database. This we have already discussed. Okay. Which of the following method can be used for sterilization of seeds to be used in tissue culture? Plant tissue culture. Yes. Which of the following method is used to sterilization of seeds in tissue culture? Yes. Log 46. Option A, surface sterilization using sterilization method. Yes. Sodium hypochlorite we can use typically. Yes. Sodium hypochlorite. Then where dry heat will come, where flame sterilization, where air blown through laminar flow will come. Any guesses? Dry heat sterilization, flame sterilization, airflow laminar sterilization. You have to go through this one also. Okay, this is homework for you. I will ask in the next class. Okay, this is very simple thing. Just to go on search. Okay. Next, which of the following is not used in protoplast fusion? That will be which of the following is not used in protoplast fusion? Any guesses? They are saying this will not be used in protoplast fusion. PEG, yes, we will use. Calcium, yes, we will use. Calcium plus DNA we will use. That will go and bind on the protoplast surface, yes. High pH, yes, we will use. Cesium chloride we will not use, yes. Cesium chloride will not use. Okay. Why means this will be typically used in where? Density gradient centrifugation. They will use in density gradient centrifugation. Correct. So this will be the right option. Which of the following is true about E. coli DNA ligase? I already told this one. DNA ligase will require cofactors. Okay. If the DNA ligase is E. coli 1, that will require cofactor what? ATP is required. If that is E. coli means NADH is required. If that is T4 ligase means ATP is required. This I have already told in our class itself. Okay. E. coli means NAD will act as a cofactor. Okay. T4 ligase means that will act, ATP will act as a cofactor. Now you see they have given easily here. ATP is a energy source. No. ATP is not an energy source. NAD is the energy source. Okay. It can ligate two molecule of single stranded DNA. Catalyze the formation of phosphodiester bond. Any guesses? From 3' hydroxy and 5' phosphate group of DNA strand. A will pakka it will come. Match and let me know the answer. Requires NADH as a cofactor, ATP as energy source. What will be the right option, students? What will be the right option? Any guesses? What will be the right answer? Yes, you have to answer. I will wait. Yes. Yes, D, A and C. Excellent. Excellent. This cannot be like it. So D will be the right option. Excellent. Next, which of the following statement is correct for 70S ribosome? They are asking about 70S ribosome. Which will be the correct? 70S, this can be 50 and 30. Okay. In 30S, 16S RNA will be there. Apart from that, what it will be there? It will have 5S RNA will be there, or else 23S RNA will be there. In larger subunit, 23S RNA will be there. Now you match. It as 16S RNA belong to small subunit. Yes, A is correct. Wherever A is coming, that is correct. It also have 8S. No, 8S will never come. So wherever B is coming, it will not come. So the right option is B. 5S RNA belong to 50S subunit. Yes, this is also correct. It can be dissociated into 40 and 30. No, this will dissociate into 50 and 30S. So this will also not come. So A and C is the right option. Then they have given here a question about genetic mapping. Okay. The resolution of genetic map depends on number of crossover which has been scored. Okay. Okay. Statement 2 is genetic map have limited accuracy as recombinants are more likely to occur at some points than at other. Some points than at other. This is simple question. They are asking from genetic map. Then what is the disadvantage and advantage of genetic map? Yes, the both statements are correct only. Yes, we know the resolution of genetic maps will be depend on the number of successive crossovers only. Okay, so this will be correct. Genetic map have limited accuracy as recombinants are more likely to happen at specific stages. Yes, some points. So both A and R are correct. Yes, option A is the right one. Another thing, they have given here the different sequencing technique and what purpose we are using. 2024 also they have asked the same question. This is a repeated question. Okay. RNA sequencing, Northern blotting, qPCR, microarray. They have given different techniques. Okay. What we will do, global gene expression of known gene from where we can identify. Then again, global gene expression of unknown gene. Okay. Unknown gene means we can sequence. Okay. RNA sequencing can done. So A, where A4 is coming. Yes, A for TK, A4 is coming here only. So, northern hybridization, transcript expression, qPCR, that will be used for relative transcript expression. Yes, relative transcription expression. Microarray, global gene expression of known gene, because here the known genes will be immobilized on the chips. TK, that will be immobilized on the silicon plates. That can be used for identifying the expression of the known genes. Yes. Again, same question, same type of question. Southern blotting, presence of protein-protein interactions, peptide sequencing, interaction of two transcript. How to do this? Okay, simple thing. Peptide sequencing, primarily we will do by MALDI-TOF. Yes. C4, where coming C4? Only in this option, only C4 is coming. So, this will be the right option. Yes, just to work smartly. Okay. We know peptide sequencing, mass spectroscopy is one of the major technique. Okay. Then you see southern hybridization, transgene integration we can be studied. Copy number also we can be studied by southern hybridization. Presence of protein of interest, whether our protein of interest is there or not, we can label an antibodies against our target protein. So Western blotting will be the right option for that. Interaction of two transcript, that means proteins are interacting or not. YTH is the critical technology. Okay. Abortive transduction is an example of, this is direct question. Okay, direct question. This is an example of generalized transduction. Yes, this is direct question. Next, what is the correct extended form of IBSC? I have already discussed this in our lecture. IBSC. IBSC what? Institutional Biosafety Committee. IBSC means Institutional Biosafety Committee. Okay. GESC means what? Genetic Engineering Appraisal Committee. Genetic Engineering Appraisal Committee. Okay. This definition they can ask. Okay. RDAC, Recombinant DNA Advisory Committee. Okay. Recombinant DNA Advisory Committee. These are some of the definitions or some of the expansions form you have to keep in mind. Okay. Where is National Institute for Plant Biotechnology is located? Yes, this is located in New Delhi. We know this one. Yes, this is direct question, New Delhi. GEAC, Genetic Engineering Appraisal Committee. I already told in our class itself. Genetic Engineering Appraisal Committee will comes under Ministry of what? Environment, Forest and Climate Change. Okay. Ministry of Environment, Forest and Climate Change. Okay. Okay. DBT will come, Department of Biotechnology will come under Ministry of Science and Technology. Okay. This is clear. Ministry of Science and Technology means DBT. Okay. Next, in prokaryotics, the genetic material, double standard single circular DNA is found in which region? Okay, we know membrane bound nucleus will not be there in prokaryotics. So this will be accumulated in a cluster form. This genetic material will accumulate and in a cluster form that we will know as what? That will be a what? Nucleoids. Okay, that is a nucleoid where that cluster genetic material will be present. In tissue culture technique used to produce seedless fruit. How to produce a seedless fruit in tissue culture? Yes, endosperm 3N will be there, that will be produced as triploid plants. So ploidy level, change in ploidy level, that will leads to this, will lead to produce seedless fruits. Okay, so endosperm culture. Yes, which of the following is correct for primer annealing temperature? They are asking about primer annealing temperature. Okay, they are not asking about TM. TM is temperature of melting for the DNA. Okay, this we will keep at 95 degrees Celsius generally for double stranded DNA, template DNA, for template DNA converting into single strand DNA. Okay, they are asking about primer annealing temperature. Base composition and length of the template DNA, this will never affect. So wherever A is coming, it will not come. Melting temperature of the primer, yes, this will come. Base composition and length of the primer, yes, this will also will come. Genomic DNA content of the plant tissue, this will also not come. Wherever D is coming, this also will not come. So B and C will be the right option. They are given here the different books, theory, proposed and characteristics, and they are asking about the authors. Okay. Skiff reagent, central dogma. Central dogma means it will convert nucleic acid to protein. So B will come 1. Okay, where here only one thing is coming, B1. Okay. Then central dogma chromosome, that will have histone and DNA. Yes, this is correct. UV light of 2600 Armstrong, that means 260 nanometer. Okay, so DNA will absorb that one. Skiff reagent is Felugal reaction. Yes, this is clear. Yes, D is the right option. Last question. What is the estimate size of human genome? What is the estimate size of human genome? Estimate size of human genome is 3.2 billion base pairs. Yes. 4.2 million base pair is for E. coli. For E. coli. Okay. E. coli means 4.2 million base pair will be there. Okay. If you want to solve more objective questions, okay, if you want to see topic wise mock test, if you want to work out, you can register in our mock test series. Okay. You can see that one. Okay. Thank you, students. We will see part 2 at 2:30. Okay. Thank you all.